This semester, we're introducing the concept of Molmodac challenges to help you clearly identify the fundamental questions and problems associated with your topic. This webcast describes the general format of a challenge and some strategies for developing and completing them. In the end, we hope that Molmodac challenges will bring greater focus to the project and will help you clarify the problems that your team plans to address with visualizations and text. We define a Molmodac challenge as consisting of two parts, a problem or question and a solution. Let's begin with the part you should imagine first, the problem. The question or problem should relate to the mechanism of action of your topic and refer to a variable or aspect of the mechanism that is important as indicated by the literature you've read and your chemical knowledge. For example, imagine our topic was the compound nitrite, and we were interested in the role of nitrite in the flow of nitrogen through the ecosystem. Bacterial enzymes that convert nitroalkanes to nitrite are an important part of this process. A relevant challenge question then might be, what is the mechanism of the conversion of nitroalkanes to nitrite by bacterial enzymes? With a challenge question like this in hand, the next step is to return to our literature and look for possible ways to describe and display the solution to the problem. Within your references, pinpoint key experiments and understand how they contribute to an answer to your challenge question. This process might involve highlighting or annotating the paper to make connections to Chem 332 concepts. Once you've combed your references and established a well-supported solution, the next step is to design and build visualizations that illustrate the solution. These may be Marvin Sketch or JMOL images that you build yourself, or structural files available in resources like the Protein Data Bank. To return to the nitrite example, after a little bit of searching, we come across a series of PDB structures supporting the key role of histidine 179 in catalysis. A mutation of this histidine residue to another amino acid destroys enzymatic activity. To illustrate this idea, we might place the wild type and mutant protein structures side by side in JMOL images. A separate image could use curved arrows to illustrate the specific catalytic role of histidine. We make a distinction as well between two types of Molmodac challenges, mechanism-based and visualization-based. A mechanism-based challenge does not require a three-dimensional visualization and can be adequately addressed using just a mechanism in Marvin Sketch. Visualization-based challenges, such as the nitrite challenge just described, require a three-dimensional image as a key piece of evidence. Use the literature references that you collect to inform your development of Molmodac challenges. As you're reading through literature, ask yourself, what are the key questions behind this research? What's motivating the researchers to do what they're doing? And what do the experiments that they do tell the reader about solutions to the problems that they talk about? With the challenge question in hand, think about how you can use interactive visualizations to illustrate the solutions proposed by your researchers in a unique way. Using these guidelines, you'll develop interesting challenges that can be effectively addressed by the technologies that we plan to use with Molmodac.